Thank you for standing by and welcome to the Arovella Therapeutics Investor Webinar and Q&A today, discussing the company's announcement yesterday that it has licensed a novel CAR-iNKT cell armouring technology from the University of North Carolina. All participants are in a listen-only mode. There will be an initial slide presentation that will last for approximately 20 to 30 minutes, followed by Q&A. If you have a question you'd like to submit, please feel free to type it in using the Q&A function within Zoom. On behalf of Arovella today, we have the CEO and Managing Director, Dr. Michael Baker. I'll now hand it over to Michael. Please go ahead.
Thanks, Matt. Big thank you to everybody for taking the time to listen in today. And you're absolutely right. We are very excited by what we announced yesterday, which is an exclusive global license for what we're calling our IL-12TM cytokine technology. And that was from the University of North Carolina, Lineberg Comprehensive Cancer Center. So without wasting any more time, I'll jump straight into it. I will be making forward-looking statements, so please do note our disclaimer. And I'd like to start with a quick reiteration about the highlights of the company. So we are working on our invariant natural killer T, or iNKT cell platform for short, and we're using that to tackle both blood cancers and solid tumours. A refresher is that we're taking ALA-101, that's been developed for CD19 positive blood cancers, and aiming to take that into clinic this year. Now, we believe that there are a number of challenges that have hampered the cell therapy sector, and that our iNKT cell platform is uniquely positioned to solve some of those challenges. And we have an exceptionally strong leadership team supporting the development, both at board level and management level. And clearly, just based off the announcement yesterday as well, we're continuing with our strategic acquisition approach to really building what we believe to be the best cell therapy products moving forward that give us shots on goal for a range of tumour types, and particularly solid tumours in this case. And as one of a handful of companies globally working on iNKT cells, we do see that we are positioned well for growth into the future. So I'll jump straight into our solid tumour strategy. I think this is something we've been discussing more recently, and we do think it's very important for a few reasons. But I think when you look at that number, it's quite staggering that 90% of newly diagnosed cancers will be some form of solid tumour. So there's an enormous societal requirement, and of course, we're excited to be a part of that. And out of that, there's born a pretty large commercial opportunity as well. Now, just to be very clear here for some background, when we think about blood cancers and solid tumours, there are quite a number of differences in terms of how we might tackle those, and certainly how a cell therapy may respond. So when we think about a blood cancer, it's a liquid tumour, so it's, I guess, relatively easier for the cell therapy to find, recognise, and eliminate those cancer cells. But when we think about a solid tumour, it's a very dense mass, and it's quite hostile in terms of doing its best to keep things out. It's creating its own blood supply. So it's quite a difficult environment for cell therapy to do what it needs to do. And there are other challenges like access to the tumour, which I've just described, but also having the requisite markers on the surface of all of those cancer cells. So we have what is called specificity, so they're not on healthy cells, but also uniformity, so they're on all of the cancer cells. And also the tumour microenvironment, we call it, this mass here, has a number of other cells, immune cells, that will actually start to support the growth of the cancer cell, and we term those myeloid-derived suppressor cells, or tumor-associated macrophages. So we think that iNKT cells represent a really exciting proposition when we think about solid tumours for a number of reasons, and I'll just talk through why briefly now. Now, in terms of killing, I'll just start there. iNKT cells have a number of mechanisms available to them to kill cancer cell types. So for those that produce CD1D, we know that the iNKT cell's natural ITCR will actually recognise that when it's lipid bound and trigger destruction of the cancer cells. We also, as we engineer them with a CAR, which we describe more like a missile that we engineer in, that will find a particular target on the surface of the cancer cells that we tell it to. But they also have a range of more natural killing mechanisms through direct cytolytic killing as well. So in addition to that, they do modify what we call the tumour microenvironment. It's been shown now that they can naturally home to tissues, and that they actually are capable of infiltrating inside of solid tumours. They actually block or kill those pro-cancer cell growing immune cells that I mentioned earlier, tumor-associated macrophages or myeloid-derived suppressor cells. And we also know that they can cross-prime other components of the host immune system like T cells and NK cells. So as I said, we do think that they're well positioned in the fight against things like solid tumors. So in terms of the strategies that we've developed, there are three major arms to that. The first is to license novel cancer targets, so aiming for targets that are not on healthy tissues, but produced on the surface of the majority of the cancer cell types. So we announced one of those late last year in Claddin 18.2. We're aiming to armor iNKT cells, so strengthening them and helping them to last longer in the body to do more damage against the cancer cells, and creating unique partnerships, as we've described previously through our relationship combining ALA-101 with Immugene's onCARlytics platform. So really the focus today is on our second strategy, which is how do we armor the iNKT cell platform. So I will have to introduce some scientific terms here, but the way we're doing that is by working with what we call interleukin-12, and it's abbreviated here to be IL-12, and that's what I'll use for the remainder of the presentation today. So it belongs to a particular class of naturally occurring proteins in our body called a cytokine. Now, cytokines and IL-12 have been very well studied for their use in immunotherapy because they can actually positively influence the immune system and leverage it so that we're actually utilizing it to fight against either infection or, in this case, cancer. So cytokines have been under development for quite a number of years, decades, in fact. The first cytokine was approved in 1986 for a particular form of cancer. And now there's been more than 40 cytokines studied in preclinical and clinical trials to date. Yet, despite that amount of research, there's only been 2 cytokines approved as single agents to treat cancer. So interleukin-12, as I said, is one of those cytokines. It is very potent. It's pro-inflammatory, so it induces an immune response. And it does have a multitude of functions, one of which is to increase the activation and cell killing capacity of immune cells like T cells and NK cells. And like iNKT cells, it actually plays a role in inhibiting or reprogramming some of those the immunosuppressive cells like tumor-associated macrophages or myeloid-derived suppressor cells. And in preclinical studies across a range of tumor types, it has demonstrated significant and strong anti-tumor effects, but it has had limited uptake as a monotherapy or as a single agent due to the fact that when it's free floating in the bloodstream, it does have unwanted toxicity issues. So in addition to that, using cytokines as monotherapies, people are starting to use them to provide armor to their cell therapies, which then is designed to enhance the anti-tumor activity of cell therapies as well. So this brings me to our IL-12TM, which is the technology that we've licensed from the University of North Carolina. And we know now that it enhances CAR-iNKT cell activity. So what exactly is IL-12TM? It's actually a modified version of IL-12 I just described on the previous slide. The major difference is instead of it being free floating in the bloodstream, we actually anchor it to the surface of the iNKT cells. And by doing so, we actually prevent it from being released into the bloodstream, which we expect to enhance the safety profile of the product. So the nice thing about working with something like this that sits on the surface of our cells is we actually incorporate it into the lentiviral vector that we use to manufacture the chimeric antigen receptor. So we incorporate both, which means we don't need to introduce any manufacturing changes to the process, and we don't have to relearn everything. We leverage what we've done already to date for ALA-101. So when we look at the combination of iNKT cells and IL-12TM, what we see is that the cells survive a lot longer and persist longer than cells that lack the cytokine, and I'll show data for that in a moment. We see that there's actually 10 times more CAR-iNKT cells 4 weeks after treatment in the bloodstream of the animals, and we also see that it does confer superior anti-tumor activity. So this work culminated over the basis of a number of years now and was published earlier this month in the prestigious and peer-reviewed journal Nature Communications. So for those that are interested in looking through the scientific data, I would certainly encourage you to have a look this up on Google and certainly go through that paper. Now, what are we talking about today? So as I said, it's an exclusive global license with UNC Lineberger's Comprehensive Cancer Center. And exactly that, to use IL-12TM and that construct in our CAR-iNKT cells. So we're delighted with the link between ourselves and UNC. Also, the commercial structure was quite advantageous or attractive, with very modest short-term capital requirements for Arovella, which is great. And we also have industry standard stage-gated milestones as well, payable in cash, as well as a low single-digit royalty. So what we're going to be doing is combining the IL-12TM with our iNKT cell platform and utilizing that to target solid tumors. So I think that's really important. As I mentioned a moment ago, the vast majority of cancer diagnoses will be some form of solid tumor. There are still a number of solid tumors that are really high priority and still sit as unmet needs, largely based off both prevalence, but also the poor survival rates that we still see for some tumor types. So we see that this is a really exciting addition and enhances our platform, and great that we get to leverage the manufacturing progress to date that we have already undertaken for ELA-101. And we do believe, and when we look at the data, which I'll show in a moment, you can see it's quite clear that the addition of this technology into the CAR-iNKT cells does enhance activity against solid tumors like neuroblastoma. So we're very excited by this because Arovella will be the first, and I believe the only company globally that's actually working with CAR-iNKT cells incorporating interleukin-12 TM. So a little bit about the inventor, Gian Petro. He is a world-renowned expert on CAR-iNKT cells. He was actually one of the pioneers of CAR-iNKT cell biology and was one of the first to create CAR-iNKT cell constructs. He is not just on NKT cells, but also on T cells, a leading authority. And he's developed CAR-based strategies to target neuroblastoma that's gone into patients in the pediatric setting as well. So he's a medical doctor by background before embarking on a research career and completed his postdoctoral studies at the Center for Cell and Gene Therapy at Baylor. And currently he's at UNC as a professor of microbiology and immunology, and also director of the cellular immunotherapy program at the center as well. So he's had a phenomenal career with more than 200 peer-reviewed research articles and received awards as a highly cited researcher, which means that others are taking his work and referencing that in their own as well. So I would like to talk about the data and what this means so you can get an understanding for why we're so excited by integrating this into our platform. So when we think about a cell therapy, a key factor for this to succeed is that we would like the cells to be administered and we would like them to last for a long time so that we get maximum damage against the cancer cells. And so this is what we're looking at here on the left-hand side. We're looking at when we either have the technology or we don't, what do the iNKT cell numbers look like in the bloodstream? So these are, unfortunately, they're complicated scientific outputs, but every dot that we see on these boxes, that's one particular cell. And we're looking at, over this side, this is a snapshot of immune cells, total immune cells. And over in this little rectangle here, it's actually the iNKT cells that are present in the bloodstream. So when we look here, this first top box is just CAR-iNKT cells. So there's no cytokine technology, no IL-12 at all. And we see that there's quite a low number. It's about 0.6%. Whereas if we look at CAR-iNKT cells where interleukin-12 has actually been pushed out of the immune cells and into the bloodstream, we see that there's about 5.4%. So we are seeing the increase. And when we look at our IL-12TM, where IL-12 is anchored to the membrane, we still see a significant increase at 4.6%. So this is excellent data. This is what we would like to see, that when we have the IL-12TM present on the iNKT cells, that we see that they elevate in number at the 4-week mark in mice. Now, I mentioned a moment ago that IL-12 also has had its challenges from a safety perspective. And what we can see here is, and this is why we wanted it to be membrane bound, we do not want it to be released from the iNKT cells and into the bloodstream. And that's what we're looking at here. We're looking at how much on the x-axis, how much of in the vertical axis, how much of the IL-12 is actually free floating in the circulatory system. So when we look at the first bar here, it's just iNKT cells alone. There's no IL-12, which we would expect. So we see nothing in the bloodstream. When we look at the CAR-iNKT cells, where it's pushed outside of the cell, we see it's in quite high levels. And then when we look at our membrane anchored, we see exactly what we want here, which is that it's not released into the bloodstream, which we do believe will enhance the safety of the therapeutic. So really important, great that it's doing what we want it to do, but we'd like to see that it also has the anti-tumor effects associated. And so this is a snapshot of the data that's been generated to date. But what we're looking at here, this is a particular form of a GD2 cell line. So it's a neuroblastoma cell line that expresses GD2 on its surface. And then we're treating the animals after that with CAR-iNKT cells that target GD2 that either have no IL-12, IL-12 or IL-12TM. So we have four groups here. The first one is animals that are treated with PBS in grey. And so we're looking at how many animals are surviving on the vertical axes. And this experiment goes out to about 65 days. So we can see that all the animals treated with essentially saline or salt water, they died around the 25-day mark. Animals that have the GD2 CAR without any cytokines, so no IL-12, they've all died before 50 days, which is still showing that the CAR's helping out. But when we look at in the pink line, GD2 CAR with IL-12, and then in green line, GD2 CAR with IL-12TM, we see that we have 75% of the animals alive at the 65-day mark. And again, this is just when the experiment is finalized at that point. So we're delighted that even though we can stick this to the surface of the iNKT cells, we still see that we get the robust anti-tumor activity with a product that we would expect to have a better safety profile as well. So again, as I said, just to reiterate and highlight, we do see increased CAR-iNKT cell numbers, and it's clear that this is prolonging the persistence and increasing the cell number and proliferation. So we know that it's not being released from the cells and into the bloodstream, which we expect to make the product safer. And just to reiterate, this is the first CAR-iNKT cell product with membrane anchored IL-12. And altogether, we're not surprised when we see those first few points that IL-12 is enhancing the anti-tumor activity against solid tumors like neuroblastoma. So I mentioned it earlier, but when we think about our manufacturing, and just to reiterate for those that are maybe less familiar with our manufacturing process, we collect blood from a healthy donor, and then we isolate the iNKT cells from that donor. We then engineer them with a lentiviral vector to produce what we tell it to, and then we grow them up into the billions before that ultimately be vialed and frozen. And so what we're able to do at this step, when we engineer in to produce the CAR or the guided missile, as we refer to it, we can also integrate the IL-12. And so that means that we're not having to rework our manufacturing pathway. It fits into the same process. We just have to modify one raw material input before we do so, which is great. So we get to leverage all of our expertise. Now, importantly, the technology is the subject of patents that have been filed. That was filed quite recently, so we have excellent protection out until at least 2043. The claims, composition of matter claims for a unique IL-12TM sequence, which is good. We haven't reached national phase yet, but when we do, we'll be looking to file that in most major conditions like the US, Europe, China, South Korea, Japan, Australia, and potentially others. And as I said, we are the only company now globally that's combining CAR-iNKT cells with IL-12TM. So in terms of our pipeline, we think this is, again, a really strong addition to what we're doing, particularly on the solid tumor front. So we've got a range of activities ongoing to give us shots on goal to treat solid tumors through our partnership with imaging, through ELA-101 and the oncolytics virus, as well as our Claudin 18.2 targeting CAR. And we could certainly conceive in the very near future that we'll be integrating IL-12TM into those programs as well. So we've got a very busy year ahead. It's been a great start to the year already. Certainly, we're very, very happy to have licensed this technology from the University of North Carolina. Also, some of you may have seen it around probably the first week of January that we actually announced we completed the GMP manufacture of our lentivirus for ELA-101. So exciting times ahead for this year. First half of the year, we aim to complete the GMP manufacturing of ELA-101 for phase 1. complete the prep activities for phase 1, such as regulatory submissions, and as well as engaging with clinical sites and KOLs. Before second half of the year, we're aiming to commence our phase 1 study for CD19 positive lymphomas and leukemias. ALA-105, which is our Claudin 18.2 targeting technology that's going after gastric cancers and pancreatic cancer. We're still excited to collect initial proof of concept data for those cells, and then we look to expand on that data and optimize the CAR construct so we've got the best possible efficacy before taking that construct and then using that to generate animal data in either gastric cancer and or pancreatic cancer models. And provide factoring for ALA-105 to look towards taking that into clinic. And lastly, for our iNKT cell therapy platform, we're excited to integrate IL-12-TM into our solid tumor programs and test its efficacy across a range of models. We're also excited that we'll enter into a sponsored research agreement with Professor Gianpietro Dotti's group. And of course, we're excited to confirm the activity of ALA-101 in combination with Imugene's onCARlytics platform. But I think you'll agree it's quite a busy year ahead, and looking forward to getting some of these data readouts. So very quickly on our financials, we're currently, at the start of the week, we're captured about 120 million. Our 4C came out at the end of last week, so we have about just under $5 million in the bank. So we're not looking to raise money, and delighted with the level of support that we've received from our largest shareholders and of course a range of others, and certainly looking forward to having additional shareholders join the register. Again, very, very clearly pointing out that those that understand both the risks and the reward that come with investment into a biotechnology company like Arovella. But we're delighted with the performance that we managed across 2023, and certainly looking forward to creating additional shareholder value for years to come. So with that, I'd just like to summarize, we do have what we consider a really exciting iNKT cell platform that we can immediately use off the shelf to treat a range of cancer types. Our CAR-iNKT cells have multiple anti-cancer properties, as well as their killing, their influencing on the tumor microenvironment. We're very comfortable that a product like this with improved manufacturing logistics is a great way forward and would help to provide increased patient access to therapies like this. We're delighted and looking forward to taking our lead product into phase 1 clinical trials for CD19 positive lymphomas. Now, we know iNKT cells also have a range of properties that we believe will assist in targeting solid tumors. With that, we're delighted now that we've expanded the platform again, in addition with the Claudin 18.2 more recently towards the end of last year, now with the IL-12 to enhance the platform overall. We're certainly looking forward to the next steps in the year ahead. And with that, I'd just like to say thanks again to everybody for taking the time to dial in. Happy New Year to everybody, and certainly I'd be delighted to take any questions.
Thanks very much, Michael. We'll move on to the Q&A as you've mentioned. Once again, for anyone listening or watching, I should say, please feel free to type in a question using the function within Zoom if you do have one in mind. Michael, the first one I'll just ask is, how did you come across this technology and working with the professor in University of North Carolina?
Good question, Matt. So we were looking, and we still are, we continuously monitor the landscape for what we would consider to be exciting targets, and then looking to see if there's chimeric antigen receptors available for licensing that for those particular targets. And that was actually how we started the conversation with Professor Dotti. And as we described more about what we were doing, we knew his background already because of his role in iNKT cells. We were pretty excited to realize that he was actually doing this iNKT cell specific work in the background. And so we ended up, I think, entering into an option agreement December 22. And so we've been able to sort of watch that scientific story develop and evolve over the last sort of 12 months or so. And it's continued to do all the right things. So we're delighted that we ended up in the position we are today, where we executed on the license agreement.
Thank you. The next question I have is, neuroblastoma would appear to be a suitable candidate for the technology given the lack of approved therapies, rapid progression and low survival. Would this be an attractive first indication for Arovella? And can you comment on whether there is a rare disease and potentially open to orphan drug designation filings?
Yeah, it's a great question. And a lot of the preclinical data does actually utilize the GD2 CAR. So we do think that's a good target, and there is some early clinical data for a GD2 targeting product from another company, Athenex, that unfortunately went into administration. But the data was looking encouraging for those young patients with neuroblastoma. Absolutely, it's a rare condition, so you would likely apply for orphan drug designation. What we'd need to do is make sure that we're sufficiently comfortable that we've got a GD2 CAR that has unencumbered access for us to work with, so there would be no freedom to operate issues and so forth. So initially for now, we'll be looking more towards our Claudin 18.2 CAR and then potentially expanding with the Imugene Oncolytics ALA-101 collaboration.
Thank you. The next question is, does the science say that the CAR-iNKT IL-12 is a better immune therapy than CAR-iNKT and safer because IL-12 is contained?
Yeah, that's still a hypothesis that we will explore, obviously, in clinic. But what's known is when you try to use interleukin-12 or IL-12 as a systemic therapy, the levels that you need to reach in the bloodstream to get a therapeutic benefit are too high, so you end up with unwanted safety issues. So ours is a very different approach. We're not necessarily using IL-12 as the therapeutic itself. What it's doing is it's amplifying the CAR-iNKT cell response and enhancing that for us. Now, we think to be prudent, having it linked to the iNKT cells will likely be a safer product. And so that's the strategy that we're committed to working with, provided we can get the data that supported it would work as well as the cells where the cytokine is actually released.
Thank you. The next question is, when will the company say that they have completed and standardized the manufacturing process?
Yeah, that's something we're still working on at the moment. It takes most of our time and resources for the manufacturing of ALA-101. As I said, big step forward was getting the GMP lentivirus manufactured over the Christmas period, and we announced that early this year. And now we're working towards finalizing the process and scaling up, which is moving in the right direction. So we don't have an exact answer for that now, but we think we're getting quite close.
Thank you. The next question's a bit of a two-part one. What is the status of the Imugene ALA-101 program, and will Arovella incorporate IL-12 into this trial?
Yeah, so as we noted last year, unfortunately, due to a technical issue in the original animal models, we were not able to get data that provided a meaningful outcome. So we're looking forward to undertaking those experiments again. If we're in a position that we have a construct incorporating IL-12 TM available, I think we'd likely include that in the study, but we'll wait to see and update the market accordingly.
Thank you. Recently, a case report was published that demonstrated the effectiveness and strong durability of iNKT cells cancer killing in a gastric patient who progressed on standard immunotherapies. Do you believe Arovella's iNKT cells now offer an exciting approach in working alongside or after expensive immunotherapies to increase survival in hard-to-treat cancers? And how might the Claudin 18.2 asset, given pending approval from Astellas in gastric cancer, which is expected to be $1 billion drug?
Yeah, look, I think we're aiming for it to be a standalone therapy in that sense. But of course, I think it would be a great idea to check that in combination with a range of things like checkpoint inhibitors and so forth. But before we do that, we'll need to get our in vivo data collected, analysed, and be comfortable with that. And then we'll take that into clinic, and then we'd start thinking about combination studies. But certainly the CAR-T product that's targeting Claudin 18.2 has been used in phase 1. The data is promising, and across a range of gastric cancer patients and also pancreatic cancer patients. And as I said, what we think about iNKT cells for all those reasons, that they naturally influence the solid tumor microenvironment, and the other benefits of eliminating other immunosuppressive cells, plus this IL-12 TM. We do think that that's an exciting prospect, but of course we need to capture the data to demonstrate that.
Thank you. The next question is, how many other companies have iNKT cell therapies in development or clinical trials?
Yeah, very few companies working on the iNKT cell space. So there's ourselves, Appia, Mink, and ImmunoKite. So that's it. It's a handful globally. Mink are the only ones with products currently in clinic. They're using what we would call unengineered iNKT cells, so they don't have a CAR to target a particular cancer. They're leveraging the natural properties of the iNKT cells. So I think really it's a small field, and of course I think now we are the only ones with the IL-12 TM technology incorporated as well. So again, I think we've carved out ourselves into a niche area, but we continue with things like the Claudin 18.2, with the IL-12 TM, we continue to differentiate from others in the iNKT cell space as well.
The next question is, what does the recent FDA decision to issue box warning to approved autologous CAR-T cell therapies regarding the possibility of recurrence of cancer mean for allogeneic therapies such as that of Arovella?
Yeah, so, and for those of you that aren't familiar with it, what we saw, I think it was 22,000 patients dosed with the currently approved autologous CAR-T products. About 22 of them had secondary T-cell lymphomas. Again, if they're all related to the CAR-T product itself, is still not conclusively identified, but at least some it looks like they were. And so what that means is there'll be some safety warnings, or what we call black box warnings, associated with those particular drugs, so that people can be fully aware of the risks associated with those particular therapies. Now, because our therapy is what we call off the shelf, at some point after the cells have done their job inside the patient and eliminated the majority of the cancer cells, we would expect the cells to be eliminated. So there are no approved allogeneic cell therapies for the cancer types that we're going after, or certainly CD19 positive lymphomas, leukemias. So there is a smaller data set to draw upon to see if those things could transpire. I think it's very unlikely with an allogeneic cell therapy, because the cells will ultimately be removed from the host that receives them. So less worried about it for allogeneic cell therapies, and I still think for the auto products, it is low numbers. I think we need to be very clear about that, and the products are still providing a lot of benefit and help to a lot of people.
Michael, and the final question is there any preclinical data assessing in a tumour-free mice environment what is the persistence of these IL-12 TM cells?
It's a good question, actually. So I'm going to answer it in a slightly different way. If we were to give an animal a particular form of cancer, and then we used iNKT cells that had a target that does not recognize that cancer with IL-12 TM, we still see that the numbers stay quite high. So we know it's driven from the IL-12 TM, not just recognition of the cancer itself that drives the increase in number. And again, we think that's a good thing because we want as many of these cells, irrespective of if the tumor cell's present. As I said, Mink, another iNKT cell company, are developing iNKT cells without a CAR at all, because they believe they still will have an impact in certain solid tumors. So the more cells we have in general, and the more we can maintain them, the better. So we don't need the CAR there for that to happen. Sorry, the target.
Thanks, Michael, and thanks to everyone for the questions. Michael, I'll hand it back to you just to provide a closing comment.
Thanks, Matt. I'd just like to reiterate my thanks to everybody for taking the time to listen in and for the excellent questions. We look forward to giving you updates as we progress. Thank you.